We're going to be focusing on the introduction to the concept of hydrogels, as well as going a little bit in depth into physical hydrogels. So first of all, this is some suggested reading material. I have posted all of these papers in the track site. And in addition to that, I recommend that you read a section of the biomaterial science book that focuses on hydrogels. All right, so first of all, what are hydrogels? Hydrogels are three-dimensional hydrophilic polymer networks that can absorb huge amounts of water, from about 10% of the dry weight of the polymer system up to thousands of times the dry weight of the polymer network. These networks are made typically from hydrophilic polymers, and I put an asterisk there because while most of the system has to contain hydrophilic polymers in order to make it a hydrogel, there are some hydrogels that are made with copolymers that might have certain hydrophobic sections. And those hydrophobic sections might actually be important because they might provide the ability to create hydrophobic or crystalline domains that act as crosslinks. But for the most part, the hydrogel, most of the polymer system of the hydrogel will need to be made of hydrophilic polymers in order to be able to interact properly with water and be able to absorb water. So anyways, these networks are made from hydrophilic polymers, which could be homopolymers, copolymers, or macromers that are cross-linked in order to form insoluble systems. And this is important. So hydrogels are insoluble. They are cross-linked, and so even though they are very hydrophilic and they might absorb a lot of water, these hydrogels will swell, but they will not dissolve unless the cross-links are broken. These hydrogels are held together through crosslinks. Those crosslinks could also be thought about as junctions or tie points between the polymer chains. And those crosslinks could be of chemical or physical or of a combined chemical and physical nature. On the right-hand side, I'm showing a graphic that represents a polymer network in trying to show you that a hydrogel could be made from covalent crosslinks, which are indicated in this particular case with these dark circles. Those could be crosslinks that are made either by a single carbon from which you have four bonds going into different chains. Although most of the times these covalent crosslinks really consist of small or medium-sized molecules, such as like polyethylene glycol oligomers that are bifunctional or multifunctional that allow you to bridge several chains. We'll talk about more examples of those crosslinks on the next section, which are chemical hydrogels. You could also have crosslinks that are made from physical junctions, and in this particular case they are kind of shown with this not that last one, sorry, that are made from this open circles, and those indicate just physical junctions, which could be either just entanglements, or they could be things like hydrogen bonding, ionic interactions, or electrostatic interactions, or other non-covalent interactions mainly. And in addition to that, we can also have like physical entanglement between chains, for example here, and loops that might actually also act as crosslinks, because just like when you have a knot, that knot will prevent the individual chains from drifting apart. Let's see. Another thing that I want to mention is that hydrogels in general are non-homogeneous. Even if you have a chemical hydrogel, for example, you're still going to have physical crosslinks whether you want to or not, because of the chain entanglement and the flexibility of the chains themselves. You're likely to also have loops. you're likely to have unlinked ends that basically have a single chain that is not actually attached. And you might have multiple crosslinks between strands. And we can actually see this on the next slide. So here we have images of what would be typically considered a ideal hydrogel here on part A. That ideal hydrogel would basically have a number of chains, and each of the chains would only cross-link with another chain in one point. So for example, this chain crosslinks once with this other horizontal chain, and once with this one, and once with this one, right? That would be an ideal scenario where you get more of that kind of cross pattern between individual polymer chains. Notice that we have this label of MC, M sub C. That's the molecular weight between crosslinks, and that's basically considered, you know, the chain molecular weight in between tie points or junctions, which for an ideal hydrogel would be a little bit easier to interpret, and it would be more homogeneous like this. Now, in reality, when you make a hydrogel, you don't have control at the molecular level, so you're going to end up with a more disorganized structure where you have maybe multiple time points between two chains, as shown here, like here and here, for example. You might have physical entanglement, as is shown right here, where you have two kind of independent systems that end up being cross-linked through this loop, basically. On D, you have unreactant functional groups, which in this case are shown by this little circle. For example, that could be a carboxylic acid or an alkyne or amine group or a methacrylate group that did not react with the intended target. And so you would end up with that functionality that is kind of just hanging loose, if you would. On E, we can also see chain loops, where a chain might actually end up looping, forming kind of interesting architectures. So again, most of the times you would end up with a very inhomogeneous system because you don't have control at the molecular level. Now, there's a lot of research into utilizing different chemical synthesis methods to try to get more homogeneous hydrogels, but a lot of times, you know, that might not be necessary because most of the times what we need is simply a porous polymer network that is hydrophilic, that would have the right chemical and physical structure to be able to carry out a function. So we don't necessarily need a homogeneous system. All right, so hydrogels could be classified in many different ways depending on how we look at this, right? One of the ways is to look at hydrogel classification from the cross-linking standpoint, or how the hydrogels are formed. And in that regard, these hydrogels would be would be classified into either physical hydrogels or chemical hydrogels. Physical hydrogels are those that are held together through non-covalent interactions. Those non-covalent, and I should mention that those physical hydrogels are sometimes called complexation hydrogels because they're being held by complexes. Those complexes could be things like hydrogen bonding. For example, you could have a DNA cross-linked hydrogel where hydrogen bonding between base pairs is what actually keeps those chains together. You could also have hydrogels that are being held together through hydrophobic associations. For example, crystallite formation between hydrophobic chains would be an example of that. You could have hydrogels that are being held by supramolecular assemblies, and we will be talking about those in a second. But these are basically interactions, typically hydrophobic interactions between these macrocycle structures and hydrophobic moieties. And then affinity complexes, where we might be utilizing the affinity between, let's say, an antibody and an antigen, or an aptamer and an antigen, or a biotin avidin interaction, and we'll discuss those in a second. But all of these are reversible and they do not involve chemical cross-linking or covalent association. On the other hand, we have chemical hydrogels, which are those that are being held through covalent bonds. And of course, there's a number of ways to be able to generate those hydrogels. You could start with monomers that are polymerized in the presence of a cross-linker, or you could begin with pre-polymers that are linked through chemical reactions. And again, we'll discuss more examples of those in a little bit. You could also classify hydrogels according to the network structure. So, for example, the hydrogels could be categorized as amorphous if they do not contain crystalline regions. And those would have to be held through covalent links or through one of these physical associations that we mentioned here. Now, we could also have semi-crystalline hydrogels where there are crystalline regions, where those crystalline regions might actually act as the cross-linker, because you could have several different chains that are coming together in certain regions, forming those crystallites. And the crystallites is basically the tie point that ties those independent chains together. We could also classify hydrogels according to their composition, depending on whether they are made from a single type of polymer, a homopolymer, or a copolymer system, or we could talk about interpenetrating polymer networks, or IPNs. We will discuss more about this at the end of the section, but basically these interpenetrating networks are a hydrogel system that are made either through the sequential polymerization of one monomer in the presence of an already polymerized polymer, or they could be made concurrently using two different monomers that have independent reaction mechanisms that do not compete with one another or affect one another. So you end up basically with two polymer systems that grow around each other and that are not chemically linked one to the other, but are intertwined one within the other, and they end up being cross-linked in a physical way, but within themselves, each of the polymers is cross-linked chemically. Again, we'll look at those later on, actually giving you some examples. The next classification of hydrogels would be according to the network charge. We would have neutral hydrogels as well. We could have ionic hydrogels, whether they're anionic or cationic, and we could have ampholytic hydrogels, which are those that have both positive and negative charges. And finally, we could have hydrogels that are either degradable or that are stable, that are not going to degrade.